CLL has been very exciting over the last few years, and at the meeting this week we've heard some really interesting data for various aspects of the disease. So first of all, ibrutinib, we've seen the first results of a randomised frontline trial for ibrutinib versus chlorambucil, the RESONATE2 trial, which showed a highly significant improvement in progression-free survival, which was the primary endpoint of the trial. But also, despite the pre-planned crossover from chlorambucil, patients progressed to ibrutinib, an overall survival advantage, which I think was somewhat unexpected. The drug's very well tolerated, and it certainly leads the way for our future and current frontline trials of targeted treatment in CLL. So the RESONATE2 trial included patients who were previously untreated but required therapy and were considered unfit for arabine-based intensive chemotherapy. So the patients had to be either over 70 or over 65 with a comorbidity. And the patients, 269 patients, were randomised 1-to-1 between ibrutinib monotherapy or chlorambucil monotherapy, and the ibrutinib continued until progression, whereas the chlorambucil went up for up to 12 cycles or 12 months of therapy. In the ibrutinib arm, at the latest follow-up, 87% of patients remained on ibrutinib therapy, so it was very well tolerated. Side effects were predictable. We saw some atrial fibrillation, about 6% of patients, which we see across the trials, and then some bruising, mainly mild, and some hypertension, but really predictable and manageable side effects. The other drug that's of interest is idelalisib, which is a PI3 kinase delta inhibitor, and we saw results of one of the first randomised trials comparing chemoimmunotherapy, so bendamustine rituximab, with or without idelalisib, and that showed a significant advantage for the patients who were randomised to the idelalisib-containing arm. This was in relapsed refractory disease. I think there's still a little bit of doubt regarding whether there really is synergy between chemotherapy and these targeted treatments, and certainly the B-cell receptor antagonists, because we didn't have an idelalisib-only arm in that trial. There's also some anxiety recently over the side effects we're seeing with idelalisib. So we always knew that there was an issue with colitis and autoimmune problems, but recently with other trials we've seen some infectious problems, and so it's clear that we have to manage the side effect profile of idelalisib very carefully, but when we do that we see really good outcomes for patients. So the 115 trial included patients who previously had treatment but were suitable for chemoimmunotherapy treatment, so bendamustine rituximab, which I think is becoming a standard therapy in relapsed CLL. Patients were again randomised 1-to-1 between bendamustine rituximab or with or without idela or placebo. And the primary endpoint of the trial was progression-free survival, which showed a significant advantage for the idela-containing arm. One of the challenges for CLL has been the speed at which the therapy is moving. So when we designed the 115 trial, the logical randomisation was as is in the trial, but because the treatments moved so rapidly, we really probably should have had a third arm in the trial to look at the difference between bendamustine rituximab plus idela versus idela plus rituximab, and that was, for understandable reasons, when we designed it, we didn't include that arm in the trial. So I think we're having to look at our trial design with the speed of movement of the trials to allow for future proofing of the trials and including all the comparators that might be important. So one of the challenges we have in terms of the new therapies for CLL, where patients take it continuously, and there's a crossover from the conventional treatment to the new treatment, is that confounds overall survival. So we and others have been looking at how can we allow for that crossover and get the true benefit in terms of survival. And one of the articles, one of the abstracts in the meeting this week has looked at that for idelalisib for the 116 trial, which is the original licensing study, and tried to use statistical analysis to allow for the crossover and see the true benefits as if crossover didn't happen. So those sort of analyses are, I think, important going forward as we get more effective treatments. The key things in terms of toxicity with idelalisib and with ibrutinib are selecting the correct patients. So we need patients who need these therapies, and certain toxicities we might want to avoid. For example, patients with previous autoimmune diseases we may not want to consider idelalisib for. We have to look at prophylaxis. So pneumocystis carinii pneumonia is an issue, and we have to monitor for CMV with idelalisib, and we have to be very vigilant in terms of educating our patients to declare the side effects that they see. So diarrhea can be effectively managed if it's treated early, but if you leave it for weeks or longer, then the patient can be unwell by the time they come into the clinic. So there's a lot of education that we need to do with the patients. One of the other abstracts, which was from my group in Leeds, was one of the challenges, I think, is with CLL is how do we combine the targeted therapies together, and how to avoid resistance, and how do we understand the mechanism of resistance that allows a logical combination. So we've been, Sarah Farag in my group has been growing cell lines which are sensitive to ibrutinib and to other targeted therapies, and then trying to, and effectively growing out resistant subclones, and so then we can look at the molecular switches that in a cellular system allow for resistance, which we can then logically combine the targeted therapies together to avoid that resistance. And of course we're applying that to the patients. So, fortunately at the moment there are relatively few patients who are resistant to these agents, but they will emerge, and we need to be prepared for that and understand the mechanism of resistance. The key, I think, is going to be the duration of therapy, and can we get patients cured, or at least in very deep remissions, so we can stop the therapies. And the likelihood is that will be combinations of targeted therapies that target different aspects of the disease pathophysiology. And we have a number of agents that are suitable to be combined together, and the trials are now started, certainly the early phase trials, looking at the combinations. We have to be very careful about safety, because we may see different side effects when we combine different targeted therapies. So I think that's the first step, and then the second step is identifying the patients who really need the targeted approach. So we know that, for example, with FCR chemotherapy, we cure a small proportion of patients. So can we identify those patients and treat them with FCR, and can we then stratify the other patients to effective treatments, which personalise the treatment towards the patients? The UK CLL forum session with the beer section, this was the second year we've had that, and it was a really good summary of the state of the art, really, of the biology and treatment of CLL. So we had three really excellent speakers, Jan Berger, Arnon Cater and Anna Shu from Oxford. And we heard about the interaction with the microenvironment between the CLL cells and the various microenvironment cells, the molecular switches that are subdividing CLL into various groups, and how that may in the future be used to define specific therapies. And I think it really showed the exciting times, I think, that are ahead for CLL, patients with CLL, and how we might treat the disease more effectively. The message going forward from this conferences and the conferences over the last 2 or 3 years is that the future for the treatment of patients with CLL, current treatment, has changed dramatically. We now have really effective therapies, and each meeting we see combinations or new treatments coming along. And I think it's really important that we continue to include patients where appropriate in clinical trials, because it's moving so quickly that's the cutting edge, if you like, of treatment. And we need to gain evidence rapidly so that we can move towards a more curative strategy for patients with CLL. In the UK, we have a really, I think, good platform of trials, both from the early phase 1 and phase 2 combination studies, leading through to big phase 3 trials. And so we have some combinations of targeted therapies, for example, ibrutinib with venetoclax, ibrutinib with obinutuzumab, and the IDELA trials are ongoing. And I think those are really leading to a lot of science that we're doing around those samples. So, for example, whole genome sequencing through the Genomics England program, which Anna Shu is leading, proteomic analysis, and then putting all that information together to look at mechanisms of response, best combinations, and avoiding resistance. I think that what we are learning is that the treatment is changing really rapidly. where as we treat patients earlier on in the disease, we get better outcomes, and the future is going to be combinations of therapy, which will lead to, I believe, eradication of detectable disease and potentially coming off treatment, and we should be aiming for cure.